Hello, my name is Lorena Pantano, and I want to talk to you today about a big project I was working for the past 3 months or so. And imagine the situation where you have multiple collaborators and they send you different type of data that you analyze, your team analyzed, RNA-seq, single-cell RNA-seq, ChIP-seq, any type of genomic data, methylation data, and they ask you to cross all these projects. Like they are studying a common system and they want to say, okay, I have this project and my collaborator have this other project, this person did this other thing, and we want to find these genes that are the key for this particular biology. And of course, they will use different species because sometimes they work with human, mouse, rat, or any other system that they have. So you want to do this right away. Like you want to give them something that even maybe you don't need a person to do this, although AI helps a lot now. But it doesn't need to be end-to-end platform that you need to develop everything, or you need a team to develop everything. Of course, you can do that if you have the budget. That would be awesome. But if you don't have that, this is a solution that we came with for this particular situation. So what is the reality of what is happening? Well, let's start with, you have different folders. Imagine that these different blue boxes are different data in the human, mouse, and so on. All these live in different folders. Now, without the infrastructure, you will go and open your Visual Code, RStudio, whatever, use AI, Claude, Copilot, and say, okay, here are all the files, please give me a script for X, Y, and Z, and you will have it done pretty fast, and then you will send the email back. But then they will ask for this other thing or this other data set. It doesn't scale pretty well, and I don't know if you want to be doing that all the time, to be honest. So as the time goes, collaborators comes to the project, studies like keep happening. So ideally, what mainly they are asking is, can we have a place where we have all the experiments and all the genes information in all the experiments? And again, you are restricted about budget. So what do? This is an example. So we made 3 decisions: composition, standardization, and automation. So basically, this will be the full picture, and I will zoom in for each of them. But first, you need to choose tools that exist that do 80% of what you need. So if we talk about genes and visualizing data, of course, we can say that IGB web app, I am a big fan of that, will do 80% of that. And it's very intuitive. People who are not the bioinformatician and computational biology can learn pretty fast how to work with IGB and what the data means. So we picked IGB, the web app version, and it's an interactive genome browser. And you can jump gene to gene easily, right? You know what is the deal here and show different data types. So basically that is what we needed, but of course there were some gaps. But at least you don't need to build a new visualization or a server-side visualization type of app solution. So that saves a lot of time, right? Second, standardization. So again, IGB, if you standardize all your output to IGB compatible, the majority of data we have will be big, graph, BAM files, BED files. The only one that were trickier are the tables coming from differential expression from different analyses. But table at the end, they are genes or region-centric, and they have a value. That region have a value, or is differentially expressed significant or not, or the region is significantly expressed or not. So you can convert that actually into a bed graph. And basically, we build a script, of course, with the help of AI. But what we did is, okay, if we have differentially expressed regions or genes, we want to just color code the bed files into this region is going to be upregulated, one color downregulated, another color significant, low fold chain, another color, or not significant. And you can play with that, whatever you want to show. And so that solves actually all our problems, because then we don't have any other data that is not supported here. And not only that, but of course you can get this script to be cross-species compatible. So you know the system that you work with, the genes names are going to be very particular for each of them, so you can guess the origin species and convert everything to human. So everything will be human genic-centric platform. And of course the third one is automate, because I have been only talking about the specific steps, but yes, you need to automate everything and deliver this somehow. So how this works is the middle gap. So this is the input, IGB is the goal. Some transformation happens, so we can transform everything. So what is in the middle is that, of course, I assuming that you have access to everything. The second thing that you will need is an AWS S3 bucket. In our case, you can use other backend, but this is what we have available. So we will upload the data there privately if you need to. In our case, it's private because this is an ongoing collaboration, so things are not still public, but at some point can be, of course, made public. And the moment that you put it here, there are like steps here that I will go in, but all these steps will happen and then it will be available in IGB. So there is really not a lot of human intervention more than new data comes, deposit it there. Next step that for now is manual but could be automatic, and that's all. So let's go into the technical part. Once you have AWS S3, the data, as well you have IGB, because IGB is a static app, so really doesn't need a server to be generating the pages. The few things that we added was because we needed to offer this to the public, is that the only thing that we need to add is CloudFront here. So CloudFront will connect to AWS after everything is well set up, and it will offer this to the public. The one thing here that we did is to have a step in the middle that is AWS Lambda function that check for user password, and that way it's private for now, but whenever we want to make it public, then we remove this step and then it's going to be public to the scientific community. Okay, so now we know that we need cloud storage. We can use CloudFront, right, to publish. We can use Lambda to protect, but still we need to do this conversion, right? So that is pretty cool, but how that looks like before getting into the last step of automating that, how this look like. So if we go, this will be the basic IGB. What is going to happen is that you are going to have different projects here. For instance, if we go to project 1, you will have 2 specific data here and you can select them. You can go to other projects, and this is a bed file, so you go to bed file and it will load that, and you can see, then you can like do what you do with IGB, you go into specific regions. Like here there is a significant one, it seems, but very easy to add data. You can add all the information you want in different columns to make it easy to select, but that is how it looks like. So let's come back about how you make this the final step, because right now I haven't say how this get updated. So basically you can create a GitHub action, because what is all the computing that is happening is very light, and the main computing is happening is if you have CSV, you want to convert it to bed file. So GitHub action is gonna go, it's gonna read from the bucket and see what is new. If there is something new, if it's a CSV, it's gonna do the mapping. If not, then it will just note that there is new files, but there is nothing to be done there. And then it will update the registry that IGB uses. After updating the registry, there is like fixing the MIME types to make sure IGB reads properly the files and CloudFront serves those files properly. And then it will update the registry into S3 with the bed files, and then that automatically get into IGB. The moment that step is done, then we are good. And this right now it could be a schedule, right now it's manual, but it could be anything that fits your setup. And that's all actually. So AWS, GitHub, some scripts, CloudFront and Lambda, you need it to be private, but after all that, this is a system that is self-update as data comes. I just want to mention that there is this public repo here where the template of all the files I use without the information linking to our setup, but at least it has the main information about how to deploy the first time, what to copy. It has the GitHub workflow, so you have it there right away. You will need to set up the secrets and all the variables that you need, some example data, and the IGB is self-contained here. So it's not like a press a button and it will be available, but definitely there is a lot of done already, so you can work from there. And that's all. Thank you very much. Bye.